Hey, my dear fellow monkeys. This video is part of a series, This Week in Science. These two things seem very important to me. The first one is about cancer research, specifically in the domain that I'm very interested in, and I have some expertise in that as well. It involves gene regulation, not necessarily based on the genetic makeup, but certain factors that enhance or deregulate certain genomic regions. The first one is by a group of researchers who have been studying super enhancers. Super enhancers are areas in the genome which have the ability to recruit a lot of proteins, and when they recruit a lot of proteins, they fold on themselves and go to a particular gene that they want to activate, and when they go near the promoter region of a gene, these proteins that are located on the super enhancers start promoting the activity of the gene that they are targeting. So what they do is they have a lot of these activating proteins which increase the activity of a particular gene. This results in a lot of protein production. It might be complete protein production, or it can be truncated protein, which have a lot of variations. The super enhancers have been shown to have certain effects in the cancer phenotype in a lot of cancers. They themselves are not necessarily involved in gene activity or activation, but they are involved in recruiting these proteins which are the activators. There is an ongoing debate about the super enhancers and their status as super enhancers. A group claims that they are normal enhancers which also do the same activity as a super enhancer but at a lower level, while the other group claims that they should be classified as separate or as super enhancers because of the fact that they are able to recruit a lot more protein compared to normal enhancers. The super enhancers have a critical role, especially during embryonic development, because the presence of certain super enhancers in a particular cell gives the cell that identity. So a super enhancer in a cardiac cell or a cell destined to be cardiac cell enhances or activates particular genes that give the phenotype for the cell to be a cardiac cell. So they are very, very important in delivering the identity of a cell during embryonic development. The embryonic cells are usually stem cells, so they have the ability to become any type of cell given the particular type of enhancers or particular type of genes that are active during development. The logical deduction certain scientists made is that cancers use cancer stem cells to propagate their population and to spread to different locations. So they might hijack the super enhancer mechanism in cells or stem cells that are present in human body, especially the pluripotent stem cells, and they have the ability to hijack that super enhancer system and start replicating the genes that the cancer cells want to be expressed, which results in a variety of things ranging from intratumoral heterogeneity, which means that the genome of cells in a particular cluster of tumors or in a particular cluster of tumor are completely different. We don't know how the lineage developed. So that is called intratumoral heterogeneity. And super enhancers have been shown to have effects in all the steps of cancer propagation, ranging from tumor initiation to metastasis and spreading to a different location. A clinical trial is underway to block the activity of CDK7, which is cyclin-dependent kinase 7. This is a major protein involved in cell cycle regulation, and it promotes cell division. So a trial to block CDK7 in super enhancers is being undertaken right now, and if it succeeds, genomic medicine targeting the super enhancers will be a reality in the near future. The major problem that is present here is that identifying super enhancers from genomic data is based on algorithms, and these algorithms predict certain super enhancers accurately while there are off-target or false positives, you can say. Also, the assays that people use in different labs is different. There is not a standardized procedure to understand how super enhancers work, and that might result in variations in how people report certain super enhancers in articles. This is a major step in the right direction towards targeting genome or the gene activity to regulate cancers. The next report is an observational study. The article is about the silent epidemic of Hepatitis B virus spreading in Sub-Saharan Africa. A lot of people are targeting diseases like HIV, tuberculosis and malaria in the developing world, which is a very good way to counteract certain preventable diseases in childhood or early in the developmental stages. But hepatitis B is a preventable disease if people are vaccinated at birth, but it is being ignored, although it is mortality from hepatitis B is higher than TB or malaria or HIV individually. The major reason that hepatitis B has been neglected is because of the fact that it is chronic and it doesn't overtly show symptoms. There is a stat that says the number of incidences of hepatitis B is around 257 million, while only 25 to 27 million are actually diagnosed. In the developed world, this has been taken care of in a pretty good manner, where most people at birth are given the hepatitis B vaccine. There are foundations like Against Malaria Foundation. This ties to my the other series of videos that I make, which is model philosophy, in which I'll also be talking about effective altruism. I also made a book review on effective altruism by William McCaskill, which I'll link in the iCard in this video. This disease claims more lives than malaria, while there are effective charities like Against Malaria Foundation which take care of malaria. There are not a lot of organizations that work on hepatitis B prevention. Although there is a vaccination-centered charity called Gavi, which focuses on vaccination of kids in the Sub-Saharan Africa. The problem with Gavi is that Gavi administers HBV, hepatitis B vaccine, along with other vaccines like diphtheria, pertussis, tetanus and influenza. But it is given at the age of around 6 to 8 weeks, while administering hepatitis B vaccine at birth or during pregnancy to the mother has the ability to prevent or at least make child immune to hepatitis B virus. Usually the mode of transmission from the mother to the child is during childbirth. This is a vaccine that can be incorporated, and certain effective charities can take this ahead, although there are charities that are doing it. A bigger role from the developed nation to the developing nations would be a better way to reduce the incidence of hepatitis B. The impact it would create is huge because of the fact that the QALYs, QALY, which is quality adjusted life years, that a vaccination would give is huge when it is given to a just born baby. This is a wake-up call for charities to take up hepatitis B vaccination drives in Sub-Saharan Africa, especially also in other countries from the east of China. The incidence rate is higher. You can see the map in the article that I'm linking in the description below. The links to these articles are in the description below. Take a look at it if you have some time. Leave a like if you like the video and subscribe to my channel for more content like this on science. Share this with your friends and family so that when they make donations, they can make to effective charities. Or if they're interested in understanding how super enhancers work in cancer biology, they can take a look at it. I've been Kaushik from The Thinking Monkey. I'll see you all next time.